Mendel, alleles, and Punnett squares. Probability in Punnett squares. You may not be able to predict what your offspring will have for certain, but you can figure out the odds. When Mendel performed a cross with pea plants, he carefully counted and categorized the offspring. He had plenty of data to analyze. He saw that when he crossed two plants that were hybrids for stem height, meaning a capital T and lowercase t, we'll talk about what these mean shortly, about 3/4 of them were tall and 1/4 of them were short. He found that the principles of probability could be used to explain the results. Now, a probability is the chance something will occur. If you flip a coin, you have a 1.5 chance of flipping heads. There's only two outcomes. You're either going to flip heads or tails. To flip heads 3 times in a row, you'd multiply 1/2 3 times. So 1/2 times 1/2 times 1/2, which comes out to 1/8. You have a 1 in 8 chance of flipping heads 3 times in a row. That is probability. Now, the way in which alleles separate during gamete formation is as random as flipping a coin. For example, if you have a gene with 2 alleles, and that is capital T and lowercase t, this is the example we're using. So when we say the way in which alleles separate, we're talking about the alleles right here that are the traits. So if we have a gene with these 2 alleles, capital T and lowercase t, it's random if the gamete will carry a capital T or lowercase t. It's not going to carry both. Now, there are both dominant and recessive alleles here. We have a dominant, which is a capital T. We have a recessive allele, which is a lowercase t. The gamete will carry one or the other. So when we're talking about gamete cells, we're talking about sperm or egg cells. That's what we're talking about when we mention gametes. Now, in a minute, you're going to see F1 and F2, and that refers to the generations. So let's look at the image on the next slide to talk about this a little more in depth. So here we have some alleles. We have capital T and lowercase t, and we're crossing with a plant that is also capital T, lowercase t. These are the plants in the first generation. Now, we said the gamete cells can only carry one allele. And if we look here, we can see that this plant, this tall plant, is either going to give off a capital T or a lowercase t. The plant doesn't get to decide. It is random during gamete formation, which are the sex cells, sperm or egg. Now, if we have this gamete get passed on, we have carrying the capital T, and this one over here carrying our capital T, we have 2 capital T's. Because we have at least one dominant trait, we know it's going to be tall. And we call this homozygous dominant because there are 2 capital T's. They are both the same, and we call this homozygous dominant because they're both dominant alleles. Over here, we see the capital T gets passed on and crossed with the lower case t. So here we have a capital T and lowercase t. The plant is going to be tall, and that's because it carries at least one dominant trait. And we call this heterozygous. There is no such thing as heterozygous dominant or heterozygous recessive. It's just heterozygous. If we look at the third plant over here, same thing, capital T, lowercase t. We also call this heterozygous. It's going to be tall because it's carrying at least one dominant trait. Now over here we have homozygous recessive. This is going to be short because there is no dominant trait. There are 2 recessive traits, and because there's no dominance here, it's going to be the other outcome, which is short. So we call this homozygous recessive. Homozygous because we have 2 lowercase t's. Homozygous recessive because there's no dominant traits. Over here, heterozygous. Over here, homozygous dominant. And this is all, these 4 plants are all part of the second generation. All right, now probabilities predict the average outcome of a large number of events, not a small number of events. Now, if you flip a coin twice, you are likely to get 1 head and 1 tails. You might also get 2 heads or 2 tails if you flip it 2 times. To get the expected 50-50 ratio or the 1 out of 2 ratio, you would need to flip a large number of times. The larger the number of offspring, the closer the results will be to the predicted value. If an F2 generation has 4 offspring, it may not match the ratio of the Punnett square. If it has hundreds or thousands, then the ratios match pretty closely. Now, genotype versus phenotype. Phenotype is the physical characteristics of an organism, meaning this is what it looks like. If you're talking about the phenotype of somebody and you're talking about the eye color, what their eye color is, such as blue eyes, brown eyes, green eyes, hazel eyes, whatever their eye color is, that is the phenotype. It's what you can actually see. We also have genotype, which is the genetic makeup of the organism. Those are the traits that they are carrying. You cannot see what they are carrying. You can only see the phenotype. Now, there are two types of Punnett squares. There's a monohybrid cross, mono meaning one, and you're just crossing one factor, like we did in the previous illustration. We saw that we were crossing a tall plant with another tall plant. We were only crossing for height there. We also have what's called a dihybrid cross, also known as a super Punnett square, and that's crossing more than one factor. So how do alleles separate when more than one gene is involved? So that's what we're going to talk about now. So it happens through independent assortment. Now, Mendel wondered if the gene that determines the shape of a seed also affected the color. Well, he set up a dihybrid cross to find out. He set up a cross between dominant, and here we have homozygous dominant because we have two capital R's and two capital Y's. And the R's refer to whether it's round or wrinkled. The Y refers to whether it's going to be yellow or green. Because we have at least one dominant trait for R and one dominant allele for Y, it's going to be round and yellow. This is homozygous. So he set up a cross between the dominant and recessive. And here we have two lowercase r's, two lowercase y's, which means they're going to be wrinkly because we're not carrying a dominant trait. And they're going to be green because we're not carrying a dominant trait for yellow. So he set up a cross between those two. Now he found that dominant traits do not always stay together and they segregate during gamete formation. Now, when we say they segregate during gamete formation, what we mean is they're actually going to separate during the sperm or egg formation. Now, independent assortment helps account for the many genetic variations observed in plants, animals, and other organisms, even when the parents are the same. For example, you may have a younger brother or sister. You guys look somewhat similar, but you may have different eye color, different hair color. You have the same parents, but you look different. And that is because of independent assortment. All right, so getting close to the end here. So here is what he set up. This is what we call a dihybrid cross or a super Punnett square. So here's what we have here. We have a capital R, lowercase r, capital Y, lowercase y. And over here in the key, it will tell us what it means. Capital R, dominant allele for seed shape, meaning if we have at least one capital R, it's going to be round. We have lowercase r, which is a recessive allele for seed shape. We need two lowercase r's for it to be wrinkled. Because we have at least one capital R, just like we said, it's going to be round. Capital Y, dominant allele for seed color. If we have at least one capital Y, it's going to be yellow, and that's what we have here. Two lowercase y's, we would see green, just like we do over here. So here we're crossing two yellow peas. Notice we're not always going to get yellow peas because we can pass on either the recessive allele for color or the dominant allele for color. Same thing over here. And here we see 1, 2, 3, 4. There are 4 chances to get green. Not only are we talking about green, we're also talking about the shape. So this is a super Punnett square. So there 9 out of 16 offspring are going to have capital RY. They're going to be round and yellow. 3 out of 16 are going to be round and green, meaning they're going to have at least one capital R and two lowercase y's. We have 3 out of 16 that are going to be wrinkled but yellow because two lowercase r's and one capital Y. Or it's going to be 1 out of 16 of them are going to be green and wrinkled. All right, so Mendel's 4 principles. Mendel's principles of heredity observed through patterns of inheritance form the basis of modern genetics, and they are this. The first one is the inheritance of biological characteristics is determined by the individual units called genes, which are passed from the parents to the offspring. The second is where 2 or more forms or alleles of the gene for a single trait exist, some alleles may be dominant and others may be recessive. That means talking about the capital and lowercase letters. In most sexually reproducing organisms, and here we have sexually reproducing, which means there's going to be sex cells involved, and you're going to need at least 2 organisms. In most sexually reproducing organisms, each adult has 2 copies of each gene. one from each parent, the mom and the dad. These genes segregate or separate from each other when gametes are formed. And remember, gametes are the sex cells, either the sperm or the egg. And alleles for different genes usually segregate or separate independently of each other. Now, further research done by other scientists verified Mendel's work. So this is a short introduction to Mendel and Punnett squares. If you have any questions, you can always leave a comment below, and we'll see you guys next time.